Hello, everyone, and welcome to this conversation. I'm Vijeta Raghuram, Associate Editor at I Wonder. For those of you who are joining us for the very first time, I Wonder is a science magazine for middle and high school teachers. It features writings about the many dimensions of teaching and learning of science in class and outside it. The magazine is published twice a year in English, Hindi, and Kannada. If you are a practicing science teacher, we invite you to write for us. To do this, please send your idea in about 100 words to iwonder@apu.edu.in. If you would like to receive updates of each webinar and a free print version of each issue, subscribe to us by following the link displayed in the description of this video. You can also catch us on Facebook. To enhance our engagement with our readers, we invite authors of an article in I Wonder for a live discussion on the second Wednesday of every month. Today, we are in conversation with Yasmin Jayathirtha. Hello, Yasmin.
Hi, Vijeta, and everybody else.
Yasmin wrote an article on testing for COVID-19 for our special issue on the pandemic in October 2020. She is a chemist and chemistry teacher associated with the Azim Premji University, Bengaluru, and she admits to be fascinated with the question, how do we find out? Thank you, Yasmin, for joining us today.
Thank you for calling.
I'm very curious to hear more about your fascination with this question. But before we start, let me give everybody a brief overview of the format of today's discussion. For the first 30 minutes, Yasmin and I will explore her article a little deeper to get more insights from her. To all of you who have joined us today, please type your questions in the chat box. We will draw your questions into our conversation in the second 30 minutes of this webinar and seek Yasmin's insights and response to them. Okay, so Yasmin, shall we get started?
Yes. Yeah. Why don't we do that?
Yeah. So, let's start with your favorite question. How do we find out? Please tell us about it.
Yeah. And why is that my favorite question, actually? See, when I learned chemistry, I learned it as a series of facts. But then for me, it was always there but how did they find that out? And what did they do? And so, in fact, I wrote the article because when the COVID-19 started and the tests started coming, I wanted to find out and I started reading. So when they said write an article, I said, okay, may I do that? Because I've already been reading about this so much and trying to find out about it. Trying to find out what is behind something, I think, looks at the process of science rather than just the content of science. And then you suddenly realize, oh, yes, it's related to other things and so on. And having been, right now you introduced me as a university teacher, but I've been a school teacher for a very long time. And there it's far more important that you kind of tell the young students, what is behind what they're learning so that it doesn't become a meaningless set of facts. So that's why.
Thank you, Yasmin. I think you have just provided a great introduction to today's theme, which is about testing and especially how we use testing in the classroom setting. Now, while your article focused on testing for COVID-19, let's start by trying to understand the meaning of testing in science. Why do we test, whether it is to diagnose a disease or in a chemistry lab? And how do these tests contribute to the learning of science and the scientific process in a classroom?
Yeah, actually, see, obviously, we all know that we test for the presence of something. I mean, we think there is something present here and we want to test, whether, say, it's a water sample or whether it's a blood sample for a disease and so on. We can also test for the absence of something. We want to say, okay, is it possible that this is not here? in which case there might be problems. So, in some sense, if we look for presence of something, we are looking to see the cause of whatever we want to find out. When we are looking for the absence of something, we kind of want to rule out something is the cause. They're subtly different from each other, they're not quite the same. And then I was thinking about what are we looking for? We are trying to trace something specific. And so before we do a test, we have to figure out what test to use, under what conditions it will work. And if it's a new test, testing for something new, will it actually get that? And we'll come on to that a little later when we talk about uh, tests by themselves. Now I'm just talking about the nature of testing. I was also thinking in some ways that tests allow us to structure what we are searching for. Now, if there are a million tests to be done, whether it's in chemistry, whether it is in disease, whether anywhere else. But if we start at random and just do all those tests, we don't get a picture of what is. But suppose, for instance, a doctor gets a group of symptoms and trying to make a diagnosis says, maybe it is this, let me test for that. If I in the chemistry lab I'm trying to have a sample that I'm going to look at, I need, at some point I can't do all possible tests and then try to figure out what it is. It's very wasteful and it'll give me so much data that I won't know what to do. So in a way, like the doctor with this diagnosis says, it could be these possible things, let me test. One also looks at, in the lab, one looks at it is these things. And I think tests also help you, first of all, to structure what you're searching for. And then it tells you the way to go later on. They raise new questions. They answer some questions. They can raise new ones. And so testing is very important. And then to come, yeah, and to a large extent, we have to, well, I suppose, determine that we can't say something is absent, totally absent. We can only say, I cannot detect it. With the tests that we are doing, with the instruments that we have, this is below the limit of detection. It really doesn't mean that it is completely absent. I don't think we can ever say that.
Mhm. Yeah, you raised some very good points now. Um, I especially liked what you said about not ruling out negative tests, and even negative tests have meaning to them. Now, when we introduce a new topic in the classroom, okay, let's say a structure of an atom or the structure of an animal cell, we usually start by uh, talking about its various features and various properties, and maybe even have a diagram showing all of these components. But it is these features that help us even design the tests, right? Like you said, what might this be? Uh,
Yeah.
so, adding this question, how do we test for a system, could make a classroom quite interesting. And
Hmm.
as a long-standing chemistry teacher at a school level yourself, you must have used tests and testing to evoke wonder and curiosity in science learning among your students. Can you share some of your experiences?
Yeah, I can tell you some anecdotes. But children love testing, even very young children. I mean, I will be talking about 8 and 9 year olds when the projects that they do. For one thing, there's the thrill of doing tests and then suddenly finding a result which you didn't expect or which you didn't know you could look for. So some of the projects that I do, and one of the projects I do for an introductory, because they all want to do chemistry, and they come in and they're very young, they're 8 and 9. So one says, look, just like in a library, you can classify chemicals. And one of the ways we classify, like you say, what do you do in the library? And they go fiction, non-fiction, then by author, and so on. And said, one of the ways we classify chemicals is as acid, base, and neutral. And how do we test? So pick out all the things marked acid, add the indicator to that, and what do you find out? So they find that out. Then we move on to food tests. And that's very interesting because the many times that I have done it, it has never played itself out the same way. It's so in one of the classes, for instance, they looked at it and said, "Oh, something." I think they were reading, and it said all washing. They got a lot of material from stuff, soap. They got shampoo. They got toothpaste, everything. And somewhere, I think I made the comment, taking from the textbook, that you know what? Supposedly, all cleaning materials are basic, so then they tested Sikakai. And suddenly it turned out to be acidic. And I said, "Oh, that's surprising. I didn't know that." And they looked at me and said, "You didn't know that?" And I said, "No, I didn't." So then, so that was something that was there. Are there other cleaning materials which are acidic? Then again, other thing which said all acids are sour, taste sour. And I said, I won't let you taste hydrochloric acid and things like that, but you can taste acetic acid and so on. Then they said, if everything sour is acidic, what is the taste for anything basic? So I said, I don't know. They say that some basic things are bitter, but I don't know whether there's a universal one. So immediately the question came, can we test bitter things to see if they're basic? And they got Karela and they did it. So you can see at this point, the tests are opening up a way of looking at the system. And they're telling you where to go. They're telling you, and this is what I mean, that bitter gourd didn't turn out to be basic. It turns out to be acidic. Another group found out that they looked at it and said, you know what, all foods are either neutral or acidic. There's no food that is basic, which again was a very interesting comment to make. The other thing we found out, and this again goes to the idea of testing itself, we made flower indicators. And in that, as they made flower indicators, we said, okay, what color does it change in acid, base, and neutral, and wrote it out on the board. Then they realized that some of the flowers showed a different color for acid neutral and a different color for base. The others might have shown one color for acid and a second one for neutral and base. It was very few that showed three different colors. So then at that point, we started talking about what makes a good indicator. That something which will not show you the difference between acid neutral or neutral base is not as good as something that will show you the difference between acid, neutral, and base, because that's more discriminatory. And this flower indicators for me, and this is just an aside, is a psychological test. The more dominant people in the class take the brightest of colors, the bright yellows and oranges, and it never changes color. And it's the ones who are left with, I only have this pale pink and it looks so dull. And when they add it, they'll get this beautiful pink and this beautiful green. And I see it every time in the classroom. So for me, there is a test out there. Now, who is the dominant person in the class who grabs the brightest colors? And they get rather disappointed. So, yes, and it helps with that and confirming a hypothesis and so on. So, I've had a lot of fun. And even trying to find out, as we said again, when we looked at the limits of testing, they say, can I test chili powder? Then I said, will it dissolve in water? Because when we add an indicator, we can only test something that is dissolved in water. So we don't know anything about chili powder because the test doesn't work for it. So we just can't do just any test and then say what result are we getting. We have to choose the tests that we do.
Actually, this concept of limits of detection is this is a very important aspect to bear in mind, and this brings me to my next question. Right in the beginning of the article, you define two characteristics for a test to be useful. specificity and sensitivity. Could you elaborate on these terms and what they tell us about the nature of science, especially its uncertainty?
Yeah. And sensitivity and specificity are really, in some ways, for biological tests. We have a different terminology for chemical tests, and I will come to that also. So, if we're looking at specificity, so look at the COVID-19 virus, for instance, to take that as an example. It belongs to a family of coronaviruses and they're related to each other, which means that they share features in common. Now, all of us get colds. Coronaviruses are flus, colds, very common around the place. All of us get colds. All of us might have that virus in our body. So, how do we know that it's COVID-19? And that is specificity. Now, if it is something 100% specific, nothing is 100%, let us call it 99.9. RTPCR comes close to that. If it's 100% 99% specific, that means that out of 100, only one would be what is called a false positive. That uh, we'll get one false positive when we do 100 tests. We might get other true positives depending on the number of infections there are, but we will get on an average one false positive, which means that it is not COVID-19, but it is some other coronavirus, which is why you have to choose what you're testing very carefully. And here they pick the way all the tests seem to depend upon the spike, which is unique to the COVID-19 virus. And they worked on that. The sensitivity, we want to find out what is the limit of detection. There it is, the limit of detection. So when you have a very sensitive test, it will pick up every positive test that there is, which means that there are no false negatives. You might have the virus, but if it's not showing up in the test, that's a false negative. So sensitivity will pick up everything. Again, RT-PCR, because it amplifies whatever material is taken from the swarm and goes on amplifying it till it's detected. It's supposed to be the raw thing. If what we consider positive in RT-PCR can be considered a true positive, what is considered a negative in RT-PCR, and we have set the limits there. How many times do you amplify before it shows up? Will be called a true, where was I? I said true positive, right? So will be a true negative. A negative will be a true negative. Now, if we take another kind of test, which is the rapid antigen testing, and it is rapid, it's very fast, it will give you the result in 15 minutes. So, which means that it takes the sample and puts it on the test strip and just tests for it. So it gives a large amount of false negatives because it could be the amount of virus was not enough to be detected. But a positive on a rapid antigen can be considered a true positive. But if it's negative and if there are symptoms, or if you people have been in contact, then you would go and do an RT-PCR test. So you can see that you're fitting the jigsaw. You're kind of saying, well, it's come negative, but this person, is it really negative because this person is showing the symptoms? So can I try something else? Or as I said, once it's positive, it can be considered true positive, which is helpful. And when they did some of these rapid antigen tests, very interestingly, when you look at the jigsaw, and I'm going on to the next thing, which would be, well, let me come to that a little later. But then I said in chemical tests or other tests, which are not sensitivity and specificity are used only for these kind of diagnostic tests. In the others, you will talk about precision, which means that the number of times you do the test, do you get the same value, or is your value differing madly if you're testing the same sample? Then there is something called accuracy, which is how close is the value that you're getting to the real value that is existent. Now, they're not they're not quite the same thing, because you can be precise, but because your instruments give a particular error, your value could be a little off from the real one, which is why most values that you get, if you notice, well, not we won't look at the medical test, but the chemical test would also say that plus or minus something, which means that that is the error inherent in the method itself. And that is something we should realize, that nothing we do. This is something that I struggle to teach my students, because when asked what is the error in the method, they go, I should do it more carefully. When I'm saying no, the error inherent in the method, and you're supposed to have done it carefully, but the error lies because of the instrument itself. And it's hard for us to think about it that way. So, which is what they are.
Hmm. Yeah. What this highlights is that you cannot take any um, result as an absolute result, isn't it? There will be some caveat, there will be some detection limits of the instruments, and it's really the technology that's, in its current form, that is letting you make inferences based on the current specificities or the current sensitivity of that
Yeah,
instrument. Right.
that's right. Also, you must remember that the technology has improved. Not so much, I don't want to say improved, but so many new technologies have come in. Now, for instance, if you're looking at RTPCR, for COVID-19, the RTPCR depended heavily on the tests which had been uh, worked for the uh, Ebola during the Ebola epidemic, because people had worked very hard to determine who was positive for Ebola and so on. And they modified it, putting in the chemicals required for the COVID-19. But the whole technique of RT-PCR came in with somebody who was trying to amplify some material, small amounts of material found from somewhere. And so, and he got a Nobel Prize for it. And it was taken on to right across from determining ancestries, from fossils and bones to determining the Human Genome Project, small amounts of material to criminal forensics. RT-PCR, I mean, PCR has been used strongly. But where the other part came also with, somebody had a very ambitious idea of saying, if we can find out the genetic genome of human beings, we'll be able to say what genes. That promise has not been fulfilled. But anyway, that was the image, that if we can write down letter by letter the genome of a human being, mixture of human beings, all that is possibly there, then we can really figure out a lot about what genes control what, we can do about disease, we can tailor make medicines and so on. And as they began to do it, earlier it was a very painstaking chemical thing in the lab. You add, you separate, you mix again, and so on. And they began to automate it. So now you put in a sample, and the automation goes on and brings it out. And that's what makes it easy to do the RT-PCR test now. So the technology is coming in there. Now I've noticed that the newer tests called LAMP and so on. What is LAMP? I'm sorry, loop. Let me just look up. Loop mediated isothermal amplification. It's a kind of simpler way of putting in chemicals, just warming them once, and then much simpler than RT-PCR. Now, that is using 3D printed little vials to shove in chemicals and put them in. So you can see that if the 3D printer wasn't there, it would have been hard to get these chemicals in the places that we wanted them to be. So science has been, the progressive science has been intimately related with the progressive technology in some ways, feeding into the progressive technology and growing because of the progressive technology.
That's a very interesting historical perspective of how technologies evolve and how they find new applications with time.
Yeah.
Um, Now, moving on, an interesting aspect that you talk about in your article is how doctors and scientists use different kinds of tests to explore different questions to study and control the pandemic in a systematic manner, almost like fitting a jigsaw puzzle. How do you think teachers can use this idea or understanding in science learning? And could you share some of your own experiences with students when you used such an approach?
Yeah, one of the ways that we can look at it, the COVID-19 was, in fact, a very important discovery, right? Because when people were looking at, they were testing people who were symptomatic. Then they started testing people who, I think, because tests were in short supply all over the world. Obviously, one had to gear up the manufacture of that, and you have to have very stringent protocols because otherwise, as we saw, the test is useless without that. So when tests were there, they were testing only people with symptoms. Then when the tests came out, a lot more tests were available. I mean, the thing to actually do the tests were available. Then they started testing. Let's test all the people the patient has been in contact with. Then they suddenly realize here are people who are not sick. They're showing no symptoms, but they're showing the presence of the virus. Now we realize that there are asymptomatic carriers. We don't know how infectious they are, but they carry the virus. So now that became a piece of the jigsaw. It's not enough for us to look for symptoms. We have to do contact tracing and we have to look at where it could have gone from. Now, people looked at, I think, now I'm not very clear, but I'm speculating on this. The other thing that changed in the response to the COVID pandemic was earlier they said hygiene, don't touch surfaces, wash your hands, don't touch your eyes, don't touch your nose, and things like that. Then I think they began to look at surfaces carry viruses, it's true. But people don't seem to be getting ill because they're touching the surfaces. So then, where is it coming from? And so the thing uh, uh, changed from don't touch surfaces and wash. They're still telling you to wash hands, but they're not pushing it so much. And they're kind of saying downgrading the risk from touching surfaces. I mean, people were panicking. They were getting online orders and leaving it for 3 days before touching it and so on. I mean, I don't blame them. I also would do that
And
and
all kinds of
things like
products came. All kinds
that.
of
Yes.
products also came saying, okay, this will
Yes,
kill
yes.
COVID-19
And how
virus.
much sanitizer do you use? Your hands are getting dry. And what do you touch? When do you sanitize? So, all this confusion. Then I think they've now downgraded that and said, please wear a mask when you go out to protect yourself and to protect the others in case you have been infected. Now, to me, that is science in action. That is science in action because, in some ways, I think, and I'll amplify this a little more looking at the whole of the COVID-19 response, but just to say, look at this thing. People think science, whatever science says, is authoritative in the sense that science gives you the last word on something. That is a very common way of looking at, well, the scientists say and science says. But the real nature of science is that it is tentative.
Hmm.
Says the best evidence is telling us this, and I'm going to follow this now because the best evidence that I have is telling me this. But if that evidence changes, I must also change my response. And the tentativeness with which you hold a theory, you hold not a theory, but hold a hypothesis or a model. I would prefer the word model. So the response from anytime you touch any surface, wash your hands too. Yeah, it's not that bad, but please wear a mask and stay distance from people is a scientific response. I know it confuses people, no, and they go, what do they know if they said this 3 months ago and now they're saying this, and so on. But that is where it goes. And if you look at the whole of the COVID response, it is really like, what is it that, what I would call uh, the science that is practiced now, in a way, the evidence based science that we are looking at. One is that it's global. So it started in China, and very quickly, well, there's some controversy about that, but at least people got to know there's a new virus. It was sequenced, it was followed. People are sequencing it all the time. I mean, we are looking at COVID-19 as one monolith which is coming out, and we are reading about 3 or 4 variants. But actually, there are a lot more. And the variants are happening, and variants of interest are being followed. People publish their data, and scientists make their data open. Journals make their data open. Newspapers made their data open, saying we won't put paywalls for reading articles about this because we want everybody to read it. People were critiquing each other's thing. Everybody was doing the work that they did the best. And then they were sharing that. Somebody else was picking up their data and using it to do something else. And then coupled with the tentative nature of the thing. I use that, not obviously not COVID-19, but I would like to tell any students that I now teach about maybe the what science is, to say this is what science is. And the scientific temper that we as teachers are asked to bring into our classes, I think scientific temper is not, I don't know what it is because I haven't been able to figure out what we are supposed to be doing there.
Define it.
But if I had to do something, I think it is really this. How do you hold on to a model as long as it's working? And how do you drop it the moment new data comes in? How do you be tentative? That is the true scientific temper that we need.
Yeah, these are really very important ideas that we have to, practice. We have to keep reminding ourselves that, okay, what we know to be correct now may not hold true, but this is the best solution we have. There is nothing that is correct or wrong, but there's no correct or wrong answer, but probably just a best answer.
The best answer. At this moment, this is the best answer. So I will follow. Yeah.
Yeah, I think in many ways, COVID-19 has provided presented a good example for many aspects of science. You started by talking about the importance of identifying the purpose of tests. Oftentimes, the purpose is, like you mentioned, the purpose could be whether there is something specifically present or not in a sample, say, while diagnosing a disease. But it could even be, what is there? We have a cell, we have a patient now, we have a new patient with symptoms that have not been seen before, or the causative agent is not known. What could be causing it? So, I mean, this was the kind of questions that doctors were asking when the first cases of COVID-19 came. But once they could identify the virus and find out the features present in it, like the genome sequence and the proteins that were present in it, that helped them devise tests and use old tests, use old technologies meant for other diseases and adapt them to COVID-19, and then find out a new question now. Okay, so whether the new patient that is coming in has COVID-19 or not, has this particular virus
or not. Yeah, and actually it's very interesting because, and why would you want to know whether there are 2 or 3 things there, and I'd like to take all of them because I believe there aren't any questions yet, so we can continue. So at some point, there is one that why would you want to identify COVID-19 from the other coronaviruses? Because if it goes bad very quickly, and you need to be alert to get people treated very quickly. That was one. Secondly, I think there would be, if you do random tests, you said you can test for everything, you
Mm,
don't quite know what is causing the syndrome because the tests are going to be all over the place.
mm, mm,
So you don't know what is there. And I'd like to share something very interesting. Two things which are very interesting, which I just read, in fact, because I'm continuing to read about tests. One is that CDC, I think the face of the COVID-19 response in the US is somebody called Dr. Fauci. And
mm,
he talked about the fact that people had been working on the AIDS. This is about vaccines, that the AIDS vaccines for a very long time, so they could repurpose it to the COVID-19 vaccine very quickly.
mm,
That is there. And now people are saying, once we got the COVID-19 vaccine so quickly, now how do we repurpose this to look at malaria, to look at other things, and so on? So it's going on out there. And the third thing about testing, I think, was this new side effect which is suddenly shown up. Very, very rare side effect. Because when people say, oh, it causes blood clots, and then uh, I think people haven't, scientists and journalists haven't been able to present it in a way that shows the risks.
mm,
So one of the things, there were two things there. One is that this blood clot is very rare. I think they talked about 24 million uh, vaccines shots given, and about 18 people showed the blood
mm.
clots, and
Yeah.
no, sorry, 65 showed blood clots roughly, and about 18 people died. So one of the people, it showed up in England first, and one of them who is looking at this very rare side effect, she said, we saw that there were clots, we saw that there were low platelet counts. Now we know that there is a syndrome linked to a blood thinner, but nobody had taken that blood thinner.
Mm,
I mean, not even that there was no platelet count, that nobody had, there was no reason for that clot. So then
mm,
I don't know why, and this is a new article that I read this morning. I don't know why I said do the test for the platelet counts. And when it came low platelet counts, we said, okay, this syndrome we know. We don't know
mm,
what is causing it, but we know the syndrome, we know how to treat it. And to all the other people who had the blood clots, they said, do the platelet count test, see if it is low. Then this is how you treat it because we know how to treat the blood
mm,
clots linked to the blood thinner.
mm,
So you're again seeing that sometimes you also need to say, let us test for this, even though there seems to be no reason to test for that. And the scientific temper that one talked about. Again, it's a very touching story for me because this is a doctor, I think, or she's in the pharma. And she was talking about her brother who died of the blood clots and said, it's very rare, it's very unfortunate that he died, but I'm going to tell you, I'm not so, I wouldn't have told him, don't take the vaccine. I'm going to tell you, please take the vaccine. You
mm,
don't know that more, the chances of dying are a lot more than this. And so it is that impersonality also, because when
mm,
something is personal, it takes a much larger risk than something that is remote from us. And suddenly, at that point, to sit there and say, yes, it's very personal and it's looming large in my consciousness, but the evidence is still the evidence.
mm,
That to me is also part of science.
mm. And that the doctors are trying to investigate and
Yes.
find
And
out
find
and
out.
establish a link. Uh,
Mm-hmm.
and until that point, I guess what they're trying to suggest is that the benefits of the vaccine still outweighs much
Very
much
much so.
more
Very much
than the
more.
risks.
Yeah. Even
And
if you are in the age group which
mm,
is likely to get the clots, they're still saying if there are more than 20, the chance is 20 in 1,000 that you're, 10,000 that you're going to get it, please take the vaccine because your risk of getting COVID and
Is
symptoms are much worse than you're
mm,
getting the
mm,
thing. They're trying to establish, the link has been established. They're
mm.
trying to establish the cause. I mean, in the sense that the causal link, I think, is fairly clear now. That it's very rare is fairly clear now. So it's there.
Uh, all right, Yasmin. So if we don't have any further questions, if we don't have any questions, we will wrap this up then.
Yeah,
Uh,
I think so. Mm.
uh, yeah, thank you so much, first of all, Yasmin, for
Yeah.
joining today. Um,
Yes.
and and thank you for sharing your experiences from your classroom as well. It was fascinating to hear about your students' responses and those stories about how they went on to test their own hypothesis, and you know and how tests can really stimulate the curiosity in students,
Yes, yes.
and and also inculcate the scientific temper. So
Yeah.
thank you
Mm.
for bringing in all of these important thoughts in today's discussion. And to all of you who have joined us today, oh, sorry, one
There is
second.
a question.
We have a question. Okay. Okay. You spoke about, okay, this is a question from Radha, who is one of the chief editors of I Wonder. You spoke about confusion. Sorry,
Oh,
this is
from
from Vijay Kumar Siddhappa. Um, you spoke about confusion matrix in machine learning. Machine learning is leveraged for healthcare. To your knowledge, what are the commonly used machine learning techniques for healthcare? Do you want to answer this question, Yasmin?
no,
I'm not
maybe
sure
you
if you
could
touched on machine
clarify
learning.
the question a little bit, because I didn't talk about machine learning. In
Yeah,
fact, I talked about technology, which is kind of amplifying the amount which is there.
yeah.
I don't really know about machine learning.
Yeah, I'm afraid I think machine learning is a little beyond what we can address in this particular webinar. Um, yeah, I think if there are no more questions,
Unless you meant automation.
you could take that and elaborate on that, Yasmin, if you want.
Yeah, rather says possibly.
Hmm.
So when I talked about automation, really, I think automation, see, trying to synthesize proteins or trying to break down proteins or DNA and find out the sequence, because sequencing tells us what the genome consists of and what a gene, what are the letters, and then it tells us, when we sequence some more, it tells us in fact what the mutations are. So when we are talking about the variants, we are looking at how does this genome differ from the genome which was earlier there, and in fact that is what is being traced, that which who infected whom, because this person had this mutation and the next person had the mutation, but the person who's like was in contact with a was in contact with B was in contact with B and C have a mutation, but A doesn't. So there you kind of do the tracing. To do this kind of automation requires, was very tedious, which is why people only say took years to sequence a small protein and so on earlier, and did it one in their lifetimes and so on, or even the DNA. Now, when people are looking at it, you're putting in the chemicals and the whole thing is automated. And the chemicals piece together and find out what is there and piece the sequences together. So, yes, in that sense, a lot of computers are used to do the sequences, do the best way of doing it, and so on. So, people are just plugging in the sample and then they're using the results that come out. to work with it. This has happened right across. I mean, learning for techniques for healthcare, I think most of them are using this kind of automatic way to work it out. RTPCR itself has that. It's going on amplifying what is there till it can be detected. And well, I would say computers and storage capacities, so that the computer can match sequences very easily and put them together, something that a human being would take a very long time to do. I don't know whether it answers your question, uh, but this is what I am understanding it to be.
I guess what was relevant for our discussion was how automation has helped us speed up this process. And, and not only automation, but the way rapid tests were developed has helped us speed up the process of testing, and which is very important in a pandemic situation uh, where you want to know and want to know fast.
Hmm.
So that was an important aspect about science in action
Hmm.
aspect of testing. Uh,
No, yeah, and automation helps because nobody could have sequenced this genome if the automation hadn't been there. It would have taken months to do it. And people are now doing it in days or in a
and the
day or
virus
things.
couldn't have been identified at all. It was
Identified
only
also,
because of
yes.
such
Yeah.
automations
And
that it was
I would,
possible to
yeah.
identify the
And
virus.
I wouldn't take away from the role of the computer memory in all this also, because rapid searching through the databases and so on. So
Yeah.
all of it would be there.
Yeah, yeah.
So I'm
Technology had to evolve at
yeah,
various levels,
yeah,
uh,
at various
not
levels, yes.
not only biology, but also in computer science and physics, and
Mm,
various fields of science had to develop
mm, mm,
for testing to be possible, and various other technologies that we use today to be possible. Um, I guess on that note, we will end this discussion today. Thank you to all of us who have joined us today. And join us again for our next webinar, which is going to happen on May 12th. And if you are interested in receiving a free copy of our latest issue, do not forget to subscribe to I Wonder. Thank you.
Okay, sorry. There is one more question.
Okay.
My antigen test will give result within 15 minutes. Yes. One of the things that the antigen test does, uh, it's rather like the color tests that you use for various diseases and also pregnancy and so on. So it's only looking at the sample, which it's taking the sample and swabbing it on the test strip. So which means that it doesn't need to amplify, which is why it also doesn't isn't very uh,
Sensitive.
sensitive, because it requires a large amount of the sample which is there. And then as that moves up the thing, we have antibodies which bind to that and give you a particular color. So it goes as fast as it takes for the sample to move up a strip of paper in a solution. So like if you dip a piece of paper, you'll see that it'll move up fairly rapidly. So that's what it is. But the technology behind that antigen test is very incredible. There is one strip, then there is another coated strip, and then there's one which tells you whether the test is actually worked or not, which is a completely unrelated thing, just to say that, look, this test has worked. So the technology is incredible, but the time taken is just enough for it to move up. For the RT-PCR, of course, you have to multiply and things like that. Mm.
There is
Would-
okay. There is another question. Consider a virus with DNA and another virus with RNA. Which one is more dangerous? Um, I guess we are not in a position to answer that question. Which one is
Yeah,
more
not at
dangerous? I
all.
guess
No,
having
no, I wouldn't know this at all. because it looks like both of them are there. The really don't know what.
Yeah, yeah. I'm afraid I see some comments now asking us about questions very specific to COVID-19 and about symptoms and all. Unfortunately, we are not. I mean, this is not relevant to testing one, but also we would rather not comment on it because these are comments that are best made by experts and doctors and
Yeah,
physicians.
yeah, that's right. I
Today's
wouldn't know
discussion
anything
was
about it at all
yeah,
to be able to
today's
say.
discussion was more about how testing, what testing can teach us about science and the temperament for science, and how can they encourage students to learn science and develop curiosity for it. So I'm afraid if we disappointed you, we're very sorry, but I hope you still enjoyed today's talk, and please join us next time for our next webinar, which is going to happen on 12th of May. Goodbye. And thank you, Yasmin, once again for joining us.
Thank you very much, Vijeta. I enjoyed myself.